So this chapter is going to cover a special kind of cell division called meiosis that's important for sexual reproduction that we see in the natural world. Here's an image of chromosomes being pulled apart, probably from an anther in a plant where pollen would be produced. And so you can see chromosomes being pulled apart here. And it's something that meiosis you would study more closely in plants in a Biology 2 course. So let's look at sexual reproduction. It does require, as I mentioned, a type of cell division called meiosis, which is different than mitosis. And the basic idea here is that in meiosis, you're going to start off with special cells that are diploid. That means they have two full sets of chromosomes. And when you're done with the entire process, you're going to end up reducing the number of chromosomes in half. You're going to only have one full set of chromosomes. So your learning outcomes for this section are going to be to characterize the function of meiosis in sexual reproduction and to distinguish between germline cells and somatic cells. And the soma, remember from earlier, we've seen it like in chromosome, lysosome, ribosome, comes from the same root, soma, which means body. These are basic body cells, and the germline cells are special cells that are found in the gonads. And the gonads is a general term for the sex organs, which are ovaries and testes, where gametes are produced. Gametes is a general term for the sex cells, and there's two kinds, eggs for the female and sperm for the male. Okay, and so within those organs, the gonads, the ovaries and the testes, so it's the ovary singular and testis singular, there are special cells in there. Not all of the cells are there, but some special cells there are the germline cells that start off diploid and then go through your meiosis. So your life cycle, sexual life cycle in any organism, animal, plant, fungus, has to involve meiosis at some point. And then after meiosis which reduces to one set, is going to be followed at some point in that life cycle by a fertilization where cells from two separate individuals, which is what sexual reproduction is when you combine genetic material from two different parents. At some point, that information is going to be combined through a process called fertilization. So during fertilization, you restore back your diploid condition. So here, in this diagram from your book, you have a haploid sperm cell, 1N, and you have a haploid egg. So this is the male gamete, and this is the female gamete, and the sperm fertilizes the egg, and the haploid number seems to be 3. Okay, and that means 3 chromosomes. And when you fertilize, you end up restoring the diploid cells that you normally find in the body of an animal. And here your diploid is 6. And there you can see all 6 chromosomes have been combined. And they're just color coding them, blue and red for one parent or the other. Here they color coded blue for the female. And the chromosomes you're getting from the male are red. And they come in pairs. So if you have a pair of chromosomes, each pair is going to have the same set of genes on there. Okay, so that's the big idea there. Again, somatic means the body cells. They have 2 sets of chromosomes. That's diploid. And then your gametes are your sperm and egg, only 1 set of chromosomes, which means haploid. And then so when the fertilization occurs, that term is a process, you're going to combine paternal-maternal genetic material. So the reproduction in sexual life cycles involves an alternation between, at some point in the life cycle, going through meiosis and then fertilization events. Okay, so meiosis basically goes from 2N to 1N to make a gamete, and then that one gamete can combine with another gamete made by the same process in another individual through a process called fertilization to restore that diploid condition. And then this individual goes about its life, matures, becomes an adult, and then produces its own gametes, completing the cycle. Here they show a life cycle for an animal called Homo sapiens, which is our species. And you can see here you have your haploid sperm and your haploid egg. If fertilization occurs, it produces the first cell called a zygote. That's the first cell after fertilization. You see it here on the list. Then that zygote goes through many, many divisions during development, and they're mitotic. And remember, we're diploid right now. And so all of the body cells of the child eventually growing more by more mitosis development turns into an adult, either male or female. And within the male or female, you're going to have your gonads, testes in males, ovaries in females, where you have your germline cells. And then they produce, by meiosis, they'll produce the gametes, and then we repeat the life cycle in the next generation. We'll go ahead and move on to the next section. This is relatively short, and we're going to look at the features of meiosis, the big ideas, some of the big ideas in meiosis. Then we'll go into the next section, we'll go into the details. So your learning objectives here, or your learning outcomes, are going to be to describe how homologous chromosomes pair up during meiosis. This is a key feature here, is the pairing of homologs. You remember, homologs are going to be pairs of chromosomes, and you get one of the pair from one parent and one from the other. And in humans, there's 23 pairs. Okay. And so each pair, one of them comes from mom and the other one comes from dad. And you're going to have 23 of those. Okay? And each chromosome pair has its own set of genes on there that are different from genes on another set. Okay? So looking at the features of meiosis here, let's do the second outcome here. Explain why meiosis 1 is called a reduction division. And remember, we mentioned earlier that you're going to go from 2N to 1N, and we don't have just like a mitosis. We have a part 1 and a part 2. There's going to be 2 divisions here. So meiosis is going to include 2 rounds of cell divisions, starting with the same cell. Okay, that germline cell that we talked about is going to go through 2 divisions. The first division is going to be called meiosis 1. It's going to have the same stages: prophase, metaphase, anaphase, telophase, as we do in mitosis. But during meiosis 1, we call it prophase 1, metaphase 1, anaphase 1, telophase 1. Once we're done with that, then we go through meiosis 2. In meiosis 2, you're going to have prophase 2, metaphase 2, and so on. But the first division is going to be the one that takes us from 2N, and is going to give us 2 cells that are haploid. Okay? And this is where we separate the pairs. So we're going to separate the homologs. But each homolog still has sister chromatids. The next division is going to be more like mitosis, where we separate not the homologs, but the sister chromatids. Okay, and you're still going to be 1N. But you're going to end up with 4 cells here that become your gametes. Okay. Now, another feature of meiosis is a process called synapsis. During prophase 1, the homologs, the homologous pairs, find each other through processes that are, as the case in a lot of biology, not completely fully understood yet, but the pairs do find themselves and combine. And that process is called synapsis. So that's a process, is a key vocabulary term for understanding meiosis. And when does it occur? I need you to know this. Synapsis occurs during prophase 1 of meiosis 1. Okay? And the pairs are going to pair up, and here they've color-coded them, blue for one parent. Now remember, that each chromosome made a copy of itself just before this division started during the S phase, right, of a cell cycle. So we have a normal S phase going on, and the pairs stay combined at the centromere with cohesin, a protein that keeps them attached there. But during prophase, the pairs find each other and form these proteins that form what we call a synaptonemal complex. Synaptonemal, like synapsis, complex. And the entire structure itself now includes both homologs. So it includes a homologous pair. And if this were humans, we would have 23 of these during this process. Okay, this is a key distinction because in mitosis, the chromosomes remain separate. They never pair up. Okay, and that's a major difference that's going to be important in how this process allows us to reduce the number of chromosomes. Anyhow, you can see here that this structure is made up of 4 chromatids. So for that reason, they may call this a tetrad. Tetra is a prefix that can mean 4. Okay. And then they also call them bivalents for the fact that there's perhaps because there's 2 chromosomes, full chromosomes there. So if we do a quick comparison, and I'm sharing this diagram, it's not from exactly from your textbooks, though it may be useful if you take a look at this more closely. You're going to start off with a parent cell in both cases. The difference is that in mitosis for body cells, the parent cell is just a somatic cell. Okay. And then over here, this one is a germ cell. Okay. So this is a somatic cell. This would be a germ cell. Now, both of them are going to have 2 N chromosomes. In this case, they're using a simple model with only 6, because it would be, if we were using the human example, we would need 46 of those, and it would just get too crowded in a picture here. So what we have to do is go through S, right? S does duplication. So we do that DNA replication. And when you do, remember that each chromosome, when the DNA is copied, the 2 pieces stay stuck together, and they remain together during the division, and so your chromosomes are looking like X's, right? So we draw them combined there at the centromere, where they're held together by that protein cohesin, and the chromosomes will look like this. And why? Because you have your sister chromatids are still stuck together, and they're identical copies, right? So right now, when we start mitosis and meiosis, somatic cells here, germline cells here, the process is the same. Okay, the germline cell, though, is in your gonad, your testes or ovaries. Now, mitosis is only going to go through 1 set of divisions, and notice here that we have 6 chromosomes, and they don't pair up. Each one of these chromosomes just pairs up individually along the metaphase plate. We're not going to show every stage of mitosis, because we learned that in the last one. Just know how the stages go. Anaphase, telophase, and then we have cytokinesis, and you end up with 2 cells that both are still diploid, because we separate the sister chromatids. Remember, they're exact copies that we made just before mitosis starts, right? And so your daughter cells of mitosis, you have 2, and they're both genetically identical. So that's something to remember here. Over here, during prophase 1, after we've gone through S, we've made our copies, the homologs pair up. So you can see now that we have 3 pairs in there. Okay, so they pair up, they form that synaptonemal complex. Some other processes are going to occur that I'm going to mention right now, one called crossing over. But the chromosomes line up along the metaphase plate, the pairs. Remember, over here they were individual. In mitosis, here you pair up, and instead of separating sister chromatids, we're going to separate the homologous pairs. And when you do that, you end up with half the number of chromosomes. So during metaphase 1 of meiosis 1, and then anaphase and telophase, this division here, so they show all of this above the red line here is meiosis. Meiosis part 1. Meiosis part 1, something to remember, is the reduction division, because we go from 2N, and when we're done with telophase 1, which is the last part of meiosis 1, you end up with haploid. So you've reduced to 3 chromosomes. The sister chromatids are still attached to each other. You go through a second round of meiosis, another division, but here's where you separate the sister chromatids. And so now each one only has 1 set of chromosomes. So now you have your cells that will become gametes. Okay. So in some ways, meiosis 1 is kind of like mitosis, because you're separating the sister chromatids, except you have half the number. So looking at some of the things that we talked about, synapsis, right? And synapsis occurs in prophase 1. I got this other diagram here to show 1 pair of chromosomes. Something else happens, another vocabulary word, 2 actually. 1, crossing over occurs, okay? And when crossing over occurs between the pairs, when they're combined in the synaptonemal complex, crossing over occurs and forms an X-looking structure called chiasma, and chiasmata plural. So it's actually going to occur in more than 1 location along every pair. We're only looking at 1, but remember, if this were humans, you have 23 pairs, okay? And you're going to have not just 1 crossing over, but you have many, but we're simplifying to just see what crossing over looks like. And you can see they drew one in black and one in white to represent perhaps one parent or another. Let's say this one is the male and this is the female chromosome that came from the generation before, right? Like all of your body cells have two complete sets, and you got one set from mom, one from dad. But if this is the germline cell going through meiosis to pair up, and you're going to have this crossing over occur. And what happens in crossing over is pieces of the chromosome get exchanged between the two sister chromatids right here from the homologs, okay? Not between the two sister chromatids itself, but between one chromatid from each of the homologs. So you're switching pieces of DNA, and you're switching genetic variation that occurs between parents. The parents are going to be carrying different versions of genes, right? We call those alleles. And when they swap, when you're done, you're going to end up with one of the sister chromatids that actually has some combination of both parents. So this is going to increase genetic variation, because then when we separate the sister chromatids during meiosis 2, you're going to have one sister chromatid that is not like either of the parents because of that crossing over. So that increases even more genetic variation. We call these types of processes that recombine genetic information from two different parents. That's called genetic recombination. And one way to get that is through crossing over. There's another way we're going to go over, but this genetic recombination leads to variation in the gametes so that the next generation has a different set of genes. And that's why offspring are not going to be exactly the same genetically, even to offspring born to the same pair of parents. This is why if we look at the human animal, and the same is true for plants or any reproducing organism, even offspring born to the same parent are not going to be genetically the same. And this is the reason why, like, if you have brothers and sisters, you all don't look exactly the same. Okay, so keep that in mind. Crossing over is a big one. And there's another process we'll go over in a little bit. So the first meiotic division I already mentioned is the reduction division, and that's where we're going to separate the homologs. Remember, the homologs are going to get separated. Okay. So like here, right here, we have a pair of homologs, and there would be, in humans, there would be 22 other pairs. They've all lined up on the metaphase plate, and they're going to get separated. The homologs get separated. If this was mitosis, the sister chromatids get separated. So it's very important to get that vocabulary correct, okay? Because we're going to be using that vocabulary in test questions. Okay? Now, for meiosis, the DNA replication occurred in both at the very beginning. Okay, mitosis and meiosis. And meiosis goes through two divisions, but after meiosis 1 occurs, we're not going to replicate DNA again. Okay, so that's what this means. No DNA replication between meiotic, not mitotic. This is meiosis, between meiotic divisions. The second meiotic division does not further reduce the number of chromosomes. All you're going to do is separate sister chromatids. So something quick to remember just for basic recall. Mitosis, we divide sister chromatids. That was the last chapter. Meiosis, first division separates the homologous pairs. Meiosis 2, the second division, separates the sister chromatids. Separating sister chromatids in meiosis 2 is more like what happens in mitosis. We're going to be looking in this section at the details of meiosis. So your learning outcomes here are going to be to describe the behavior of chromosomes through both meiotic divisions. Remember, there's meiosis 1, sometimes it's abbreviated as M1, and then meiosis 2. If I just say M, that would be mitosis, right? So you put a Roman numeral when you're talking about meiosis. Then your second learning outcome is to explain the importance of monopolar attachment of your homologous pairs at metaphase 1. This is key here because during metaphase of mitosis, each individual chromosome has the sister chromatids, and each sister chromatid attaches to opposite poles. But in meiosis 1, so this would be mitosis. But in meiosis 1, you're going to have the chromosomes pair up, and the entire chromosome is going to attach only to one pole. So instead of this chromosome, the sister chromatids attaching to both poles, we're only going to do monopolar. And this is going to allow us to separate the homologs instead of separating the sister chromatids. So that's an importance, right? So you're just kind of getting overall, that's the big idea there. And then number 3 is differentiate between the events of anaphase 1 and anaphase 2. And quickly, in anaphase 1, the homologous pairs separate. Okay. The homologs. So it might be the homologs separate. Okay. And then in anaphase 2, the sister chromatids separate. So that's a big idea there. There's a difference. So in anaphase 2, it's more like mitosis where you separate the sister chromatids. But in anaphase 1, you're separating the entire chromosome. Each one is a part of a homologous pair where you got one from one parent, one from the other. So now let's look here at the information on the first slide here. And your meiotic cells, these are going to be those germline cells. The ones that are found in your testes or ovaries. These are going to have an interphase period that is just like interphase and mitosis. But there's going to be one. And this interphase is going to have a G1 and then S a synthesis where we replicate your DNA to make copies of all your DNA. And then you go through G2. And then once you finish that, then we're ready to go through meiosis. Okay. So, and then you start meiosis. Now, the thing about this is you're only going to have this full interphase before you start the process. But once you start the process and you go through meiosis 1 and you go through telophase 1, when you're done, you're not going to go through S again. So we're not going to have another synthesis of new DNA or replicate DNA again. You don't have that. Instead, you start meiosis 2 right away. And remember here in meiosis 1, the big idea, you're separating, during anaphase, you're going to separate homologs. And then in anaphase 2, you're separating sister chromatids. So make sure you remember that. So looking at prophase 1, the events are very, very similar overall. The difference is going to be what's happening with the chromosomes. Okay, so what happens here? Well, your centrioles that are here are going to replicate, and then they're going to separate, start moving towards opposite poles, and you're going to start to form the mitotic spindles. In this case, you're meiotic, not mitotic. Okay. The other thing that happens here is your nuclear envelope is going to disintegrate. And so these things are all similar to what happens in mitosis. Where the big difference here is you can see right here your homologs are going to be pairing up. That process called synapsis is going to be occurring here. Okay, so synapsis occurs. You're going to form that synaptonemal complex that we talked about in the prior section there. And some very important things are going to be occurring here, crossing over. Okay. Crossing over is going to create genetic recombination. Okay, and what that means is that you get one full set of chromosomes from each parent, and you're carrying them in all your body cells. But when it makes you who you are, what you look like, behaviors, your physiology, the internal workings of the cell, your metabolism, all of that is a combination of the traits you inherit from both parents. Okay. And so each chromosome is carrying traits from each parent. Now, when it comes time to meiosis to produce sperm and egg, the individual now is going to recombine traits that they were carrying from both parents. So we're carrying traits from both parents, and they're being expressed. We kind of look like our parents somewhat, and so on. But what's going to happen when you make the sperm and the egg is we're going to recombine those traits and get different combinations of those traits, right? So that's going to occur here. And that one of the processes that helps that is called crossing over. And that crossing over is going to happen between non-sister chromatids. Okay, that's the key there. And you got to remember here that during that crossing over, we saw a little picture of that, is that pieces or segments of a chromatid are going to exchange pieces with each other, and that exchange of chromosome material is going to allow us to recombine that material in a different combination. And what's really important here to remember is that all along a chromosome or a chromatid, and then remember we have 2 exact copies before we split them up, all along the chromosome are going to be locations where genes are situated. And those are called gene locus singular or loci plural. Okay, so loci would be plural. And so there's gene loci all along there. Every segment would be coding for a different trait or different gene or different protein. And because these genes go through mutation, we end up with alleles. Alleles are different versions of the same gene. When we start covering genetics in the next chapter, we're going to look at plants. Plants go through sexual reproduction. And in a simple case here, there could be a gene for flower color. That's your gene, but you may have two different versions of that gene. So you might have an allele for purple flowers, purple color, and an allele for white flowers. It's the same gene. It's for flower color. We're talking about trait of flower color, and then you have two different versions of there, right? So depends on what you inherit, what you're carrying. You may have one allele or another allele. You remember the site where crossing over occurs that's going to end up creating this recombination here is called, it's going to create these structures called chiasmata that you can see during meiosis 1. So taking a look closely here, you're going to have, say this is a homologous pair during prophase 1, and these are sister chromatids, but if I point to one of the blue ones, which I think the color coding from my earlier slide had this as the maternal, and this is the paternal. You remember, if this is human, you're going to have 22 other pairs doing this, so we're just looking at one. And so these two in the middle, one blue and one red one, those would be non-sister. They're not identical. They're from two different chromosomes, right? So these are the non-sister. That's where the crossing over is going to occur. It's not going to occur between two sister chromatids. If we did, we'd be just exchanging exact copies of the other. So let's say now that when we go through this crossing over, that this location is going to be the location of a gene we're going to call A. Could be flower color in a plant, could be related to skin color, or some enzyme that runs your metabolism in your cell. Okay, there's hundreds to 1,000 different genes on an individual chromosome. So let's just call this gene A, and this is going to be the location. And you remember, both sister chromatids have exact same copies because during S we just made duplicates, right? And let's say this is location for gene B, and the maternal chromosome is carrying a copy we're going to label as a capital letter. So it's the capital A allele. And then the paternal one at that location is for the same trait, except they're carrying a different allele, different version. And it's lower. We're going to indicate that by lowercase, right? So remember, this is within, this is one pair of chromosomes within that individual. This could be your cells. And you have, we're just looking at one specific, two different genes on the same kind of chromosome. Remember, there could be hundreds to 1,000 other genes depending on the chromosome on there. We're just looking at two here to make things simple. And then you have 22 other pairs of chromosomes all doing this as well. Okay, so at points along there, you're going to have crossing over, and here they're just showing two crossover events. It could be many along the same chromosome, and we're going to crossing over is going to take place, and at metaphase, you're going to be able to see that there is the chiasmata. It looks like an X there, but you can see that the two locations are switching places. And so when we do separate, you're going to end up separating these chromosomes. The chiasmata disappear or get separated because we're separating the chromosomes. And you can see now that we have two chromatids that now have a recombination of the original parents. Okay, and so we think about this here. At this location right here, when we go back to what I drew over here, we have the big letter alleles. Okay. And the same over here on this other chromatid. The chromatids that I'm drawing on first right here in the anaphase chromosomes that have been separated already, they have the original complement. Big A, big B, and little, this one would be little a, little b for the paternal maternal. So this chromatid here and this one here are exactly like the original parental chromosomes. But over here, these two here are actually recombinants. Because if we think about it, the paternal segment that got separate, that got recombined here to make this chromatid is carrying a lowercase a, whereas at this location we have a capital B. Okay. Now it's going to be kind of hard for me to draw on this one here, but this one right here at this location, I'm going to come and label it up here. That's capital A, and then the location here is still going to be lowercase b. Right. So what do we get now? If we didn't get crossing over, and if crossing over didn't occur like it did here, and then we separate the chromosomes, all you're going to get are what you got from your parents, right? You're going to get big A, big B, and then you're going to get little a, little b, and little b on each of the chromatids. But over here, we're going to end up with a sperm or an egg that has big A, big B, and another sperm or an egg that has little a, little b. But over here on this one here, now we're going to get sperm or an egg with little a, big B, and then another one with big A and little b. And so here you end up with gametes. Just looking at just those two genes now, those right here, those are genetic recombinants. And so these two that I've drawn here in the middle end up being chromosomes that have some different combination from the parents before, from the parental generation, whereas these here, this sperm or egg and this sperm or egg are like the parent one. And so this happens at many locuses all along the chromosome. You can have several crossing overs, and then this is happening on every pair that's lining up, homologous pair that's lining up at the metaphase plate. Okay, so now we're at metaphase. Right, so remember that crossing over is going on in prophase 1. Then at metaphase, the pairs get finally end up in the middle. Remember, they get pulled, the chromosomes get pulled back and forth. We were talking about that in mitosis until they line up in the middle along that metaphase plate. And the key difference here is you have pairs they're paired up. Okay, and each homolog is going to be attached to one pole or the other. Okay, so your microtubules are going to attach to one of the homologs, not to the sister chromatid itself. The homologs are going to line up at the metaphase plate side by side, and the orientation of how the paternal and maternal line up is a random thing. If we look at a simple case where we have a model where you're only going to have, instead of 23 pairs, you're going to have just 3 pairs, 6 total. This chromosome right here could have lined up on this side, right? So it's one side or the other, the same for the other two, one side or the other, one side or the other. And so each one has 2 possible positions that they can line up, each pair. Okay, the way the pairs line up on that metaphase plate is random. And remember, when we're talking about pairs, you have one paternal, one maternal. So the paternal-maternal could line up one way, or they can line up another way, and the next pair of chromosomes lines up independent of any other one. So they don't influence each other, and it's purely random. This is where more genetic variation is going to come in. Okay, so we can see that here with a very simple model where we have 6 chromosomes, so 2n is equal to 6, and that means we have 3 pairs overall. In a human, this would really be 46 chromosomes with 23 pairs. But you can see here one possible way that the chromosomes can line up, and they've color-coded them. So you can get that where all maternal line up on one side and all paternal line up on the other side. Okay, that's one possibility. The opposite could happen here. Okay. And then you can see some of the other possibilities here. What you end up getting, where you have 6 chromosomes in 3 pairs, so really it's 3 pairs of chromosomes, you end up getting 8 combinations. So there's 8 combinations here. So you get 8 combinations when you just have 6 chromosomes in 3 pairs. What about a human who has 23 pairs? What are the number of possible combinations you can get up for the way they align here? Okay, so keep in mind here, we're ignoring crossing over. We're just looking at the way they line up. This is one way, and if we separate them, we get one possibility. this one right here, the way they lined up there, that's going to give you another possibility where you get this set of chromosomes and this set of chromosomes. And the point is here, you're getting different combinations of entire chromosomes now. That increases variation as well. Is there a way to figure this out? Well, at each position, there's two possible combinations. So we're going to say we're going to take 2 and we're going to raise it to the n power, and the n power is going to be the number of pairs. Okay. So if we try that formula where you have 3 pairs, that would be 2 to the 3rd power, and that's going to be 2 times 2, which is 4, and then times 2 again, that's going to give you 8, and that's why we have 8 here. But what about humans? It's 2 to the n power, number of pairs. We have 23 pairs. The answer is it's actually more than 8 million. So it's approximately 8 million possible combinations. Okay, there's no way we can draw those on here, right? But 8 million possible combinations is going to give you 8 million possible combinations that end up in a sperm or an egg, right? This is one reason why brothers and sisters don't look the same, because every sperm is going to have some different combination, and then you add crossing over in there, and now you go up orders of magnitudes, not in the millions, 10 millions. You're talking about 100 million, trillion possible combinations for the number of combinations of recombinations of your parents' genes that you inherited, right? So in anaphase 1, your microtubules spindles are going to pull apart your homologs, okay, to either pole, and we separate the homologs, right? And then keep in mind that crossing over has occurred as well, so you're going to have chromatids that have different combinations. And so what happens here, when your chromosomes are lining up at random on either side of the metaphase plate, we're going to get what's called independent assortment. And this is going to be important. You're going to want to remember this when we go back and we start talking about genetics in the next chapter. We're going to come back to when those chromosomes line up independent of each other. The main reason the main big idea here is these alleles are also lining up independent of each other. And that's going to be an important idea when we talk about sexual reproduction and look at genetics and how inheritance patterns come about. Okay. And then we go to telophase 1, and here, just like in telophase of mitosis, your nuclear envelope reforms, your chromosomes are going to decondense again, and so on. But what's going to happen here is what we're separating is we've separated the homologs from each other, and at this point here we have nuclei that are no longer a nucleus. It's no longer 2n, it's haploid, because you have half the number of chromosomes. Okay? Now cytokinesis may or may not occur. It depends on what kind of organism you're talking about, whether or not the cytoplasm separates and you get 2 separate cells, but the nuclei definitely become separate. After that, then we may have, you're not going to go through an interphase, you're not going to have a G1S, you're not going to have DNA replication occur again. Instead, meiosis 2 is going to happen. It might happen right away, or it might be a delay, but we're not going to go through another interphase. Okay. So for meiosis 2, this one's going to be more like mitosis, except we have half the number of chromosomes. And why is it going to be like mitosis? Because the chromosomes at metaphase 2 are going to line up individually instead of in pairs. We separated the pairs now. What we're going to do is, during anaphase, you're going to separate the sister chromatids. Okay. Meiosis 1 is where we separated the homologs, right? And telophase is like any other telophase, is you're going to reform the nuclear envelope, your chromosomes decondense, cytokinesis occurs, and you're going to end up with 4 cells that are genetically different now, and they have half the number of chromosomes. Okay, so this is just showing you what's occurring here, and they're doing this with a simple model where we only had 2 pairs of chromosomes to start with. So here you only have 2 chromosomes here. Same process is going to occur here, your mitotic spindles, and we have 2 cells here because we went through 1 division already. Here the chromosomes line up individually along metaphase plate, the sister chromatids get separated during anaphase, and then eventually your nucleus forms during telophase, and your chromosomes are going to start to decondense and so on, and cytokinesis occurs. You're going to end up with 4 cells, and each of these 4 cells, if we look, and they're just looking at 2 chromosomes here, you're going to have different combinations of entire chromosomes, and then the crossing over, like you see there with the different color coding there. You're going to end up with 4 gametes, sperm or egg, that are genetically different from each other because they have different combinations of alleles. So your final result of this process, you're going to have 4 cells, not 2. Mitosis gives you 2 cells that are identical. Here, meiosis gives you, when you're done, 4 cells. They're going to be haploid, and they're going to be genetically different. Okay. In animals, those cells directly end up forming gametes, either sperm or egg. Okay. In plants, fungi, and many protists, these cells may divide further. It depends on the organism, the species. And if that occurs there, then if they divide further, then they're just going to be, each individual cell as result is going to be exactly, you're just going to make extra copies of each one. And this last one here, it would require some explanation. We're going to go ahead and ignore this. Okay. This gets into some advanced plant biology we don't have time for. So, errors can occur in this process of meiosis, and an error can be non-disjunction, and the non and the dis is a double negative. To disjoin means to separate. Okay, a non-disjoin means to not separate, right? So we're talking about not separating the pairs. And so I went ahead and drew this for an organism that has a diploid number of 6 chromosomes, so that means 3 pairs. And if we go through meiosis, then we should get a haploid number of 3. Okay, so what I've drawn here is a germ cell fixing to go through meiosis, and we'll go through meiosis. During meiosis 1, you're going to separate the pairs, but I've only attached the middle pair to one pole here. And so what that means is that this pair is not going to disjoin. They're not going to separate. We're going to have an error occur here. They should all separate the pairs. Each homologue of a pair should be separated, but they're not. Instead, both of these are going to go to one side. And what's going to happen here is, when we go through the first meiotic division, we should end up already with n equals 3, the haploid number, but we don't. On this side over here, because this pair didn't disjoin, we're going to end up with n minus 1 here, which is, in this case, if n is 3, then 3 minus 1 is 2. And then over here, the other side is going to get n plus 1, and so that means 3 plus 1, and you end up with 4 over here. If this were humans, okay, then n minus 1, you would end up with not 23, but 22. And then over here, you would end up with 24, because for us, 2n is equal to 46, but n is equal to 23, right? So if we had one non-disjunction, we're going to have this error here. And so the end result here is going to be n minus 1 gametes, and over here, n plus 1 gametes. And those are going to be called aneuploids, okay? And that just means they're not diploid, they're not haploid. They have either extra or missing chromosomes. And this can be a problem because you're missing a chromosome. Okay. So let's say here, if this is a sperm here and it's n minus 1, and you have a normal egg that's n, in this case n is 3, right? And this is an egg, and this is a sperm, and this sperm comes and fertilizes this egg. Well, it is n minus 1, and this is n. So you're going to end up with, when they combine, you're going to end up with 2n minus 1. Okay, not 2n. The zygote should be 2n. We covered that in the prior sections. You end up with 2n minus 1, and instead of having 6 total, you're going to end up with a chromosome. You're going to end up with 5. So you're missing an entire chromosome, and that leads to the zygote not developing correctly and dying in the process, which might be called spontaneous abortion or miscarriage. The same thing would happen here, except if we combined an n with an n plus 1, you're going to get 2n plus 1. And if it was this hypothetical species, it would be 6 plus 1. You would end up with 7 chromosomes. You have an extra one in there. Okay. Now, it does depend on which chromosome the non-disjunction occurred, whether or not the individual survives all the way up to at least birth. In some cases, it depends on the chromosome. The embryo fetus is viable and they survive, and sometimes with severe problems, sometimes with not so severe problems. You may have heard of a common instance of Down syndrome would be a case of an N+1, but it does depend on the chromosome. So looking at meiosis versus mitosis, overall, if you were to compare them, meiosis is going to be characterized by 4 differences, 4 big things that are different. Number 1, homologous pairing. We do not have this pairing. That does not occur during prophase of mitosis. This occurs during prophase 1. Okay, that's different. Okay. Another thing that occurs here is that sister chromatids are going to remain joined at the centromeres, and then they end up getting separated during anaphase 1. In mitosis, that doesn't happen. Instead, we're separating sister chromatids. Okay. Another feature that is looking at meiosis is that the sister chromatids are going to attach to the same pole. So for each homolog, both sister chromatids are going to attach to one pole. Both of these are going to attach to the other pole. That's different because in mitosis, each chromatid attaches to an opposite pole. Okay, so this right here, this one is key here to separating the homologs from each other. And then another key feature is that DNA replication is going to be suppressed. That means once we finish meiosis 1, we're not going to replicate DNA to start the next round of divisions. Okay, so that's another big difference there. So here we're going to do a comparison of meiosis and mitosis. And your learning objectives are to describe the distinct features of meiosis. And then your second objective is to describe the differences in chromatid cohesion in meiosis and mitosis. And then explain the importance of suppression of replication between meiotic divisions. So go through meiosis 1, and before starting meiosis 2, we don't have another replication event that occurs. I found this diagram. It's extra other than what's in your book, and it compares the two side by side. For meiosis 1, this bracket should actually come out to here, starting with prophase 1, going all the way to telophase, cytokinesis, and then meiosis 2 to get your haploid cells, which symbolized by 1N. And this is for a simple model of just 4 chromosomes. So 2N would equal to 4. And 1N would equal to 2 in this case. So you can see here at the end, your haploid cells only have 2 chromosomes. If this were a human cell, it would be 46 and then 23. And so they're compared side by side. Down here is mitosis, and up here is meiosis. And here they just say, well, we don't have a 1 and 2, so we go from interphase. This doesn't mean that this is much longer time. It's just that we don't have 2 parts to this, just 1. You have your interphase where you have G1 synthesis where DNA is replicated, and then G2, and then you start prophase, prometaphase, metaphase. Remember that the chromosomes line up individually along the metaphase plate, and then in anaphase we're separating the sister chromatids. Okay, so chromatid separation here for mitosis, and then telophase, the chromosomes uncoil, the nuclear envelope reforms, and then you end up with 2 diploid cells. And there are no genetic recombination mechanisms here, so these 2 cells are clones. They're exact copies of each other genetically. For meiosis, there's error in this particular point here, but you do start off with 4 chromosomes. They go through interphase, so you're going to have G1, then synthesis occurs, and then G2, and then during prophase 1, synapsis occurs. So the chromosomes actually pair up. You do not see that here in prophase of mitosis. So synapsis occurs, crossing over, which is where genetic variation occurs. So crossing over occurs. Here in prometaphase, they should still be paired up. So that's the error that's here. If we follow what our book shows in the written description and so on, the homologous pairs should still be paired up. And then they do line up at the metaphase plate with metaphase 1, except they are lining up with their in their synaptonemal complexes. They're lined up as tetrads. Then during anaphase 1, the homologs get separated. But remember, when they line up here, each pair lines up independent of the other one, which is where we're going to get more genetic variation in there, because the pairs line up independent of each other. And so paternal and maternal chromosomes can line up on one side or the other for each pair. And we discussed how in humans that alone leads to over 8 million combinations. And then if we include the crossing over, we get other adds to that variation for each gamete produced. So then we get to telophase, the new nucleus forms, and then cytokinesis. And then so we have 2 cells, and at this point we've already gone haploid. So the first division, this one's the reduction. Okay, and we reduce because we've separated the pairs. Each pair's the homologs have been separated. Then from here, the meiosis 2 is more like mitosis because we separate the sister chromatids here. Okay, so just like down here, the way they drew the diagram, up here our sister chromatids get separated. At metaphase 2, the chromosomes are going to line up individually. If this were human, it would be 23. And remember that the spindle, the mitotic spindles, the kinetochore microtubules are going to connect each chromosome to both poles so that we can separate the sister chromatids. So they get separated, and you end up with 4 cells here, and each one of these cells it shows genetic variation because of the recombination. And so every one of these is 1N or just N, and they're haploid. Okay? And in animals, these would become sperm or egg cells. So your book does such a comparison here. It starts with the one parent cell here. If this was going to go through meiosis, then this would be the germline cell. Over here, going through mitosis, this would be somatic. And so you're going to have chromosome replication, which is your S during the interphase, and then you start your division. Here, they just right away start showing you the stages of mitosis. Remember that during prophase, the mitotic spindles form and the centrioles move to opposite poles. The nuclear envelope disintegrates or degenerates. Chromosomes coil up, and you can see them with a light microscope. They then connect to the spindle fibers and get pulled to the middle at metaphase. So they're not showing prometaphase here, but the spindle fibers connect during prometaphase. Then metaphase, they line up in the middle, and once everything checks out at that checkpoint, then the separase goes and breaks down the cohesin, and your chromosomes get separated, and then you end up with 2 daughter cells that are genetically identical, and both are still diploid. They still have the 4 chromosomes that we started with. So we start with 4 chromosomes here, replicate them, and so we have our sister chromatids connected at the centromere. For meiosis, that's different. During prophase 1, all of the things that happen here to the nuclear envelope, the spindle apparatus forming, the centrioles migrating to opposite poles. But here, remember that we're going to have synapsis occur. Synapsis and crossing over. Then at metaphase, the synapse, the tetrads stay together, and we're going to connect 1 chromosome to each pole only, rather than each chromosome to both poles. And then we separate during anaphase and telophase. We're separating the homologs from each other, and so they separate. and meiosis 1 is your reduction division. And so here we now only have 2 chromosomes. We started with 4, so we're already haploid at this point. What's left is to separate the sister chromatids, and we should go through the second round of divisions in meiosis 2, line up the chromosomes in the middle, separate the sister chromatids there, and then reform the nuclear envelopes, and you end up with 4 daughter cells that are all haploid. And when you look here diagrammatically, the way they've drawn the chromosomes, the chromosomes are drawn slightly different. They're different color, and then you have they show at least one crossover event there, just to remind us that we have 4 different genetically different. They're different combinations of the prior generation's alleles. So to focus now on some of those aspects, you're looking at the homologous pairing. That is only seen when the paired chromosomes find each other. That's specific to meiosis. Now, a question might come about, how does and they number their chromosomes based on their size and banding patterns. So let's look at chromosome 1. How does chromosome 1 from one parent and from the other, which are in our cells, every pair, how do they find each other? And that process is not really understood. Okay, so more research, probably studying that stuff. More clues as time progresses may help shed more light on that. The second point here is that the sister chromatid cohesion. There are proteins that are going to help hold them together. We talked about a protein called cohesin, which is the name of the protein causing that cohesion. So what's holding those sister chromatids together? Remember, during replication, the DNA strands stay stuck together in the middle with a special protein called cohesin. Okay. So the cohesion that occurs between the sister chromatids in meiosis is similar, but does involve a specific meiosis-specific cohesin proteins, right? So it's different types of proteins found that do that same job. The synaptonemal complex proteins, so those are different proteins. So here you have your homologous pairs finding each other. They're actually going to have proteins that help hold them together, and those proteins are the synaptonemal complex proteins. They've been identified in diverse species, and they do show a little sequence conversion and what that conservation. Sorry, and what that shows is those proteins have a genetic code for them. Based on the genetic code, will give you a sequence of proteins in or a sequence of amino acids make the protein. And it seems that among species they've studied, these types of proteins, the sequence is not strongly conserved. So that means that there's been some changes that have occurred within eukaryotes where meiosis takes place. So now looking at the centromeres of sister chromatids, they don't separate during meiosis 1. Remember, they're going to stay together. So when we talk about a pair of chromosomes, before we get to a pair, if we look at one of the chromosomes, it's made up of 2 identical sister chromatids. They're held together here. The sister chromatids represent exact copies of a strand of DNA with the proteins. And in mitosis, each chromosome lines up individually, and we separate the sister chromatids. That's in mitosis, but in meiosis 1, the pairs find each other. They're in that synaptonemal complex, but we separate the homologs from each other. So every pair, you separate the homologs instead of separating the sister chromatids like you do there in the top picture. And so just a reminder that this is a key mechanism that helps us reduce the number of chromosomes, that they pair up like this, and then we separate, they meaning the homologs of each pair. So that's a key distinction there. So so when we look at meiosis and mitosis, the sister chromatid cohesion at the centromere during all of meiosis 1 is going to occur. So we're gonna retain that cohesion, that cohesin protein there, the one that holds the 2 sister chromatids together. But we're going to lose cohesion from the arms. So what does that mean? And so along the arms you have this cohesion occurring with this with the synaptonemal complex. Now, when we're going to separate in meiosis 1, we're going to have to pull apart these. So we got to get rid of the synaptonemal complex proteins, but we have to maintain the cohesion between the sister chromatids, right, in order to do this. So how do we protect? Because the protein that holds each of the sister chromatids together, that cohesin, it gets broken down. We covered this back in mitosis. It gets broken down by a enzyme called separase. And in meiosis, a protein that's been identified called shigoshin helps protect that enzyme from being broken down during meiosis 1. And shigoshin is Japanese in origin, and it represents a guardian deity in Japanese culture. And so they named it after that, I think, and I believe it may have been Japanese scientists that discovered this. So I'd have to go back and check up on that history of the discovery of that protein. So during meiosis 1, this protein is present and protects the protein cohesin from being broken down by separase during that process. Now, this is review that separase was talked about during the mitosis chapter. So looking at now the sister kinetochores for the kinetochores for sister chromatids. Those are going to attach to the same pole during meiosis. So again, I go back and I'm just going to draw one pair. Okay. If this is mitosis, there's a kinetochore for each sister chromatid, one on each side. It's a little plaque protein on either side. And in mitosis, each chromosome connects to a microtubule at the kinetochore, and then is going to get pulled away. The sister chromatids are. But if this is meiosis 1, both the kinetochores are going to connect only to one pole, and the other one of the pair is going to connect to the other pole. So there's a big difference. What we see here on the left side, which this is for mitosis, and then on the right, this is for meiosis 1. Okay, that's the difference. Now, this is meiosis 2. Then meiosis 2 is more like mitosis. Okay, so how does that occur, right? For meiosis 1, and the basis for that, what we call monopolar, you're only connecting to one pole. Like this particular chromosome is only connecting to one pole. Seems to be having to do with the centromere kinetochore complexes. When we talk about complexes in this sort of context, we're talking about those proteins that make up the kinetochore, the centromere, and the microtubule binding to it. So there's some differences there. Obviously, it would make sense logically. There has to be some kind of difference there. However, that difference is reduced during meiosis 2, where each kinetochore connects to an opposite pole, just like in mitosis. So when it comes, so this is for mitosis here. Let me rewrite that. And then we get all this also in meiosis, but in the second meiosis, which is more like mitosis because the sister chromatids get separated. Another thing that to consider is that replication is suppressed. After meiosis 1, we don't replicate the chromosomes. So what is the mechanism that allows that, right? And so there has to be some mechanism that suppresses that replication. We replicate before we start meiosis 1, but after meiosis 1, during meiosis 2, we don't replicate. Why do we want to replicate? We don't want to double the number of chromosomes. We're trying to reduce the number of chromosomes. So we need to suppress that. Okay. Now, in normal mitosis, and there is, remember the cyclins that help regulate aspects of the cell cycle. There is a specific cyclin called cyclin B. And in mitotic divisions, that's for mitosis, right? From the last chapter. The cyclins, remember the cyclins go up and then they go back down during the cell cycle. So during mitosis, cyclin is degraded and lost. During mitosis, And that seems that next time we come back around and go through the cycle again, that allows for DNA replication. But during meiotic division, between the meiotic division 1 and meiosis 2, after meiosis 2, the cyclin B seems to remain there. And so the presence of cyclin B may be part of the mechanism that prevents synthesis from occurring, which is where DNA replication occurs. That's the connection there. So there's a correlation there, so there may be something to that. So remember, meiosis is going to produce cells that are not identical genetically, right? And what is that? The two reasons for that. One is that random orientation of each pair of chromosomes on the metaphase plate. They line up as pairs, and the way they line up on there, each pair is independent of the other one. And remember, for the most part, one chromosome, if we ignore crossing over, one chromosome is paternal, one is maternal. So in an extreme situation, one event, and it's a probability, small as it may be, any one of those orientations is possible. It is possible to get all paternal and all maternal on the other side of the metaphase plate. And then you can start playing around with every possible permutation in there. And in humans, you get over 8 million combinations because of the 23 pairs. The other one is the crossing over that occurs during prophase 1, where non-sister chromatids exchange parts with each other. And what does this result in? This results in variation in gametes, and then when fertilization takes place, that results in variation in the offspring in a population. And that's important because that variation is what selection can work on from the environment to help drive processes that are associated with changes over generations in a population, which we call biological evolution, right? So if you have no variation, then population can't change, right? And so this would be important. Where is the importance of sexual reproduction? Okay, if that wasn't for sexual reproduction, then organisms would be basically having clones. There would be no genetic variation, no promotion of genetic variation because of the random orientation of the chromosomes because of crossing over. And if you have little or no genetic variation in your offspring and the environment changes, and the environment's been changing since the beginning of since the Earth formed and life began on this planet, if the environment changes and one individual cannot handle those changes and everyone is genetically alike, then all of them are not going to be able to handle it, and your population, your species can go extinct. So if you do have variations, some individuals in there may have the right combination to be able to handle those changes, and then so they survive and produce offspring, and the species continues. So that's the big idea there for sexual reproduction.